All right, well now I'm going to begin exploring molecule construction. So to do this, we're going to start with atom-based construction. So I want you to go up to the node atom-based construction, click on it, and again it will be highlighted here in this sort of black box. Drag it across to draw the structure node, and then we now have these two nodes next to each other. We want to then take the output of draw the structure, click on that black arrow and that black triangle, and then connect it to the atom-based construction node. Next, we want to right click on atom-based construction, and then at the top here we have execute. This is going to then run the atom-based construction node based upon our input structure. In this case, it is paracetamol. So press execute and you'll see that these two green arrows appear. This means that the node is currently running. So it's performing the atom-based construction. It's now finished, so the atom-based construction has finished, and we can now look at the generated molecule. So if we right click on atom-based construction, we can look at then connected to output table. And we can see that we have generated 24,360 molecules. Now, one of the things that we learn in the lecture material is that the molecules that are generated by atom-based construction methods may have poor synthetic tractability. And we can clearly see very quickly that this is indeed the case. However, there are many that we may believe that we can make. Now the key, again, takeaway is that atom-based construction generates a lot of molecules, and we can see that there will be, if we go and look through these compound lists, there'll be many examples of duplicate molecules. This is because, for example, we have replaced the hydrogens in methyl, and we may be able to replace any of the three hydrogens in methyl with, for example, a fluorine, so we generate three molecules that are identical. They simply have just been a hydrogen to a fluorine. One of the interesting things is that, of course, we could then go ahead and score them. We will do that at a later sort of stage. But the key takeaway here is that we have replaced various different atoms, we have expanded some rings, and we generated 24,360 structures starting from paracetamol. Next up, we want to do fragment-based construction. So fragment-based construction is all about trying to find fragments to replace or to grow molecules. In this particular example, we have paracetamol, and what we're going to do is we're going to identify substructures of paracetamol, and we're going to actually replace those substructures with fragments based upon bioisosteric substitutions. So just to be clear, a bioisostere is a substructure or a group that has similar physical or chemical properties to another group. And what this means is that it enables us to have broadly similar biological effects. However, what it means is that if we have a liability in our molecule, we can easily find replacements, fragment-based replacements to our liability. So to achieve this, what we're going to do is we're going to, first of all, go up to fragment-based construction up here on the left. We're going to take this node, first click, and then drag across. We're going to then connect, draw the structure to the bottom port here. And then we're going to take the bioisosteres substitutions table. I'm going to click and drag and bring that across. And then we're going to connect read table into the top here. We're then going to go ahead and right click on fragment-based construction, and we're going to press execute. That's now finished executing. We can now look at the results. We can right click on fragment-based construction, go to the bottom here of connected to output 1, click on that. And we can see that this table is a bit different. So the first thing I want to highlight is we generated significantly fewer rows. So we've generated significantly fewer molecules. In the previous example for atom base, we had generated 24,000 different molecules, or 24,000 molecules I should say, for which there may be duplicates, but a significantly larger number. Here we've only generated 495. So there are many different columns here, and we can go through them briefly. And so the first sort of thing that I want to show you is that we have our original molecule in the first column here, and then we have this sort of R group matched column, and this is a smarts pattern, and that smarts pattern is highlighted here as this sort of R group highlighted region. So this is the sort of fragment that we're looking to try to either replace totally or replace a part of. We then have our isosteric replacement. To be able to view the isosteric replacement rather than a smarts pattern, what we can do is we can right click on that, go to available renders, and then we can go to RDKit 2D depiction. That will then show us this is our isosteric replacement that we're going to have for part of this molecule, or part of, this substructure here. So if we then move across, we can then see the reference where this substitution came from. We can also find a sort of suggested molecule. This is the newly generated molecule. And so to be able to see this in more completeness, what we can do is we can actually reduce the size of the reference, and then we increase the size of this final column here. Right click on the header at the top, and then go to RDKit 2D depiction, and we can see this is the transformation. So this was the sort of matched group, and this is then our sort of isosteric replacement, and this is the final molecule on the output. And we can see if we actually look through these molecules that in many, many cases, these molecules are significantly more tractable than the molecules that were being generated by the atom-based construction. And in fact, some of them are actually including several large groups. Next is reaction enumeration. So reaction enumeration is slightly different. So in reaction enumeration, what we're going to do is we're actually going to look to take a collection of reagents, a reaction transformation, and we're going to look to then use the collection of reagents and reaction transformation to generate new molecules. So in this example, what we're going to do is we're going to go to draw the structure in this reaction enumeration, and what we're going to do is we're going to click on reaction enumeration, and then we're going to go up to the left here, and we're going to press select all upstream nodes. We're going to, this will then highlight the nodes that are connected, and then we're going to go and drag this across here. Move ourselves across and then down, and you may notice that it's currently missing this input here. We're going to go across to the SDF reader, click on the SDF reader and drag it across, and then what we're going to do then is we're going to take the output of SDF reader and pass it into the reaction enumeration. Right click on reaction enumeration and press execute. The reaction enumeration has now finished, so we can start to look at the results. So right click on reaction enumeration, down to connected to output 1, click on that. And we can see again the table is different, but there are some key columns. So the first column is this product column, which I will make a little bit bigger. And we also have reactant 1 and reactant 2. So in the case of reactant 1, this was the reactant, one of the starting materials, and this was then another starting material, another reagent, and we reacted it together and we generated this product. And we can see that actually we have a large number of different molecules, and the basic idea is that these all follow the same reaction scheme. What that means is that these molecules should be more synthetically tractable because I have started from a molecule, a reactant, which I know I can purchase, a building block I know I can purchase, and I have applied a transformation which I know can be performed in the lab. This means that the product molecules that are shown here should also then be synthesizable. We can also see that we have generated some duplicates, and this is simply because of the fact that when we go away to a large reagent database, we may find duplicate building blocks, and that leads us then to duplicate molecules. But you can see here that we have now successfully generated paracetamol by taking initially a collection of reagents, building blocks, and a reaction, and we have successfully regenerated. So one of the attractive parts of this is that if one can buy reactant 1 and reactant 2, we should be able to make the product. This means that we can quickly build large libraries using reaction-based generative design. And reaction-based construction approaches are attractive because, of course, whilst they don't provide control, they do provide a large degree of synthetic tractability. Reaction-based approaches are often the strategy for building large virtual libraries which can then be made on plate or in the lab. And this means that we can build large diverse collections which will then be used for hit finding or data generation type activities. So that's a basic introduction to the different types of molecule construction approach. So we have atom-based, fragment-based, and reaction-based molecule construction. In the next video, we're going to explore a little bit about molecule scoring. And in particular, we're going to think about filtering and descriptor calculation.